Memory is the cognitive process responsible for encoding, storing, and retrieving information. Let's explore the three fundamental processes of memory. First, encoding is the process of converting information into a format that can be stored in memory. It transforms sensory input into a mental representation. Storage is the second stage, where encoded information is maintained over time. This can range from milliseconds to decades, depending on the type of memory. Retrieval, the final stage, involves accessing stored information when needed. This can be spontaneous or require conscious effort. Memory is organized into several distinct types, each serving specific functions and operating over different time periods. Let's look at the main types of memory and how they're classified in cognitive science. Sensory memory briefly holds sensory impressions after stimuli have ended. It lasts only milliseconds to seconds, allowing the brain to process continuous sensory input. Short-term or working memory temporarily holds information that's currently in use. It has limited capacity, about 7 plus or minus 2 items, and typically lasts seconds to minutes without rehearsal. Long-term memory stores information for extended periods, potentially a lifetime. It has virtually unlimited capacity and encompasses both conscious, explicit memories and unconscious, implicit memories. A crucial aspect of memory formation is consolidation, the process of stabilizing memories after they're initially acquired. During consolidation, memories are transferred from temporary storage to more permanent forms. This process occurs primarily during sleep, especially slow-wave sleep, and involves neural replay and strengthening of synaptic connections. Without proper consolidation, memories may remain fragile or be forgotten entirely, highlighting the importance of sleep for learning and memory. Perception and memory are not isolated processes. They constantly interact with each other. What we perceive influences what we remember, and what we remember influences how we perceive new information. Our perceptions, how we see, hear, touch, taste, and smell, form the foundation of what enters our memory system. The quality of our sensory input influences the strength of our memories. What we pay attention to determines what will be remembered. When we find meaning in what we perceive, our brains are more likely to encode that information into lasting memories. Equally important is how our memories shape what and how we perceive the world around us. Our prior experiences create frameworks that shape how we interpret new stimuli. These memories establish expectations that filter what we actually notice in our environment. Cultural and shared memories also play a role in creating perceptual biases that affect how we experience the world. A powerful example of this interplay is our sense of smell. If you have a strong memory associated with a particular smell, that smell can trigger vivid memories and emotions. Unlike other senses, smell is directly connected to the limbic system, which processes both emotions and memories. The olfactory bulb has direct pathways to the amygdala, which processes emotion, and the hippocampus, which is crucial for memory formation. This unique connection creates particularly powerful and emotionally charged memories. For example, the smell of freshly baked cookies might instantly transport you to childhood memories in your grandmother's kitchen. This constant interplay between perception and memory is essential for creating a coherent and meaningful experience of the world. Our perceptions are constantly being encoded into memories, which then influence how we perceive new information. This continuous cycle creates our unique understanding of the world around us. Without this continuous feedback loop between what we perceive and what we remember, our experiences would feel disconnected and lose their meaning and significance. Researchers are discovering that improving the brain's waste removal system can significantly enhance memory in older mice. The brain's lymphatic system plays a crucial role in clearing waste products and toxins that can impair cognitive function. Studies show that when this system works efficiently, memory function improves, particularly in older mice. This discovery offers a potential therapeutic target for addressing age-related cognitive decline in humans. Another fascinating area of research addresses a mystery that affects us all: why we don't remember being babies. Researchers are exploring infantile amnesia, the phenomenon that explains why adults can't recall memories from their earliest years. Brain development studies reveal that high plasticity during infancy may affect how memories are encoded and stored. The rapid formation of new neurons and connections during early development might actually overwrite or prevent the formation of retrievable memories. Sleep also plays a crucial role in memory formation, particularly for perceptual memories linked to positive emotions. Recent studies reveal that perceptual memories associated with positive emotional experiences are selectively strengthened during sleep. This happens during specific sleep cycles when the brain processes and reorganizes the day's experiences. During sleep, memories transfer from short-term to long-term storage through a consolidation process that strengthens neural connections. Another groundbreaking discovery involves a newly identified type of neuron that plays a fundamental role in recognition memory. Scientists have discovered a new type of neuron that is essential for recognition memory, one of our most fundamental cognitive abilities. These specialized neurons help the brain distinguish between objects we've seen before and new ones we're encountering for the first time. This discovery highlights the complex connectivity between the hippocampus and prefrontal cortex in forming and retrieving memories. These new insights into brain function and memory are opening exciting possibilities for understanding and potentially treating memory disorders. Recent research has uncovered a remarkable connection between our gut and brain, known as the gut-brain axis. This complex communication network connects your digestive system directly with your central nervous system. A key component of this connection is the gut microbiome, the trillions of bacteria, fungi, and other microorganisms living in your digestive tract. The vagus nerve serves as a critical communication highway between your gut and brain. It transmits signals in both directions, allowing your gut bacteria to influence brain function. When these signals are disrupted, it can lead to inflammation and potentially affect cognitive function. Research published in February 2025 revealed a significant connection between specific gut bacteria and memory function. Scientists found that certain bacteria are associated with improved memory formation, enhanced attention span, and better cognitive flexibility. Conversely, other bacterial strains were linked to increased risk of Alzheimer's disease, memory impairment, and cognitive fog. Your diet plays a crucial role in shaping your gut microbiome. Different foods can either promote beneficial bacteria that enhance memory or harmful bacteria that impair cognitive function. One primary mechanism connecting gut health to memory is inflammation. Harmful bacteria can trigger inflammatory responses that travel along the vagus nerve to the brain. This inflammation can disrupt normal brain function and impair memory formation. Research suggests that reducing harmful bacteria and promoting beneficial ones could significantly improve cognitive health. The emerging research strongly suggests that maintaining a healthy gut microbiome may be essential for preserving memory and cognitive function throughout life. Current research is focusing on potential therapeutic interventions for memory loss, a condition affecting millions worldwide. One promising experimental treatment is GL273, which has shown significant potential in restoring memory and cognitive function in Alzheimer's disease models. The amyloid precursor protein plays a central role in Alzheimer's disease. When cleaved by specific enzymes, it forms amyloid beta plaques that disrupt memory function. Many therapeutic interventions target this process. These therapeutic interventions work through various mechanisms to combat memory loss. Brain imaging studies reveal how effective treatments can restore activity patterns in areas critical for memory formation and retrieval. Current research includes numerous clinical trials and studies examining potential memory-enhancing drugs. Some of these, like pitavastatin and gemfibrozil, are being categorized as smart drugs based on promising results in animal models. These advancements offer hope for future therapies that can combat age-related cognitive decline and neurodegenerative diseases, potentially preserving memory function for millions of people worldwide. Recent research is visualizing brain activity during visual perception as geometric patterns. These visualization techniques map neural responses to visual stimuli, creating patterns that reveal how our brains process what we see. These geometric patterns reflect neural activity in the visual cortex, showing how different visual stimuli activate specific regions of the brain. This research helps us understand how our brains transform visual information into perception and memory. Recent studies are also developing new olfactory tests to assess people's ability to process smell. These tests evaluate three key abilities: discriminating between different odors, identifying specific smells, and remembering odors over time. These olfactory tests provide valuable insights into how our brains process smell information and how this processing relates to memory function. Both visual and olfactory research provide crucial insights into how our brains process sensory information, how this information is encoded, and how it relates to memory formation and retrieval.